Hi, good morning, everyone. Welcome to Grand Rounds. Um, I wanted to give some announcements, go over the format for Grand Rounds, and then I'll introduce today's speaker. The Grand Rounds for the foreseeable future is going to be by WebEx. Bear with us as we work out all the kinks in making that run smoothly. And thank you everyone who's been supportive and behind the scenes to make this happen. For announcements, our speaker next week is our very own Anne-Marie Albano, who will be talking about coping and managing in these incredibly difficult and stressful times. For the format for Grand Rounds, so our speaker will present in the beginning as we normally do. And then at the end, we will have a question and answer session. People can, throughout the Grand Rounds, people can type their questions into the chat function on WebEx. And the other thing I wanted to mention is that if people are having any difficulty with the WebEx interface, you should feel free to reach out to Simon Williamson, who can help troubleshoot. Simon will be away until this coming Sunday, but anytime after that is available to help. Okay, so our speaker today, we are very fortunate to welcome Dr. Eldad A. Dr. Hode is associate professor with tenure in the Department of Pathology and Cell Biology at Columbia University. Prior to the COVID-19 pandemic, his research focused on red cell biology and iron biology. More recently, his research has focused on serological testing for SARS-CoV-2 and on the use of plasma both for treatment and for prophylactics against COVID-19. Dr. Hode is the New York Hub Principal Investigator for the REDS 4P, which stands for the Recipient Epidemiology and Donor Evaluation Study in Pediatrics. He's also the recipient of several other NIH grants. He's authored more than 80 peer-reviewed articles covering a range of topics, including red blood cell biology, transfusion medicine, clinical pathology, and hematology. And so with that, I will turn it over to our speaker, Dr. Hode.
Thank you, Jonathan, for the introduction. Let's see if I can share this. Can you hear me all right, and can you see the screen?
Yep, you're all set.
Okay. So when Dr. Lieberman asked me to give the talk, he wanted me to share my experience during the COVID-19 pandemic, and so hence the title, and that's what I hope to do today. So I have no relevant financial conflicts of interest to disclose. And when I was making this talk, I was asked to come up with some objectives, I guess maybe for CME credit. And I felt that, so the objectives I came up with, the first one was to understand the role of clinical pathologists. So I am by training a clinical pathologist, and many clinical pathologists are aware that most people don't know what a clinical pathologist is. And so hopefully I'll tell you who I am, what I do, and you'll get a sense for, by the end of the talk, at least what clinical pathologists are responsible for in the hospital. And then I'll take you through the antibody testing that we developed for SARS-CoV-2 at Columbia. And I'll share with you some of the nitty-gritty. Hopefully I won't bore you too much with the details, but hopefully by the end you'll understand how tests are developed and validated. And then I'll give you some clinical correlates and an update on what we've been doing with convalescent plasma and passive immunity studies here and around the world, and a bit of future directions. I'll also mention, if you do have questions during the talk, please don't hesitate to text them in, and I'm happy to stop and answer questions if something doesn't make sense. So what is a clinical pathologist? I went to Wiki, the source of all knowledge, and the definition was is the it's the field concerned with diagnosis of disease based on the laboratory analysis of body fluids, such as blood, urine, and tissue homogenates, using tools such as chemistry, microbiology, hematology, and molecular pathology. So in a nutshell, a clinical pathologist runs the labs in the hospital. And so when people hear pathologist, they usually are thinking of anatomic pathologists. And so clinical pathologists are distinct from anatomic pathologists. You could be both. I happen to be just a clinical pathologist. Anatomic pathologists are involved with the diagnosis from organs and tissues. And so autopsies, tissues that come out of people after surgery, looking down a microscope, that's anatomic pathology. The clinical pathologists are involved in the hospital with setting policies for testing. So we have more administrative responsibilities and making sure that the labs are run appropriately and that are set up correctly. And we somewhat act as a consult service for other physicians when questions arise regarding testing. And my training happens to be in transfusion medicine and stem cell therapy, which is a branch of clinical pathology that's responsible for the blood bank and the stem cell therapy lab. We also have an outpatient unit because we use it to treat patients. And so it's one of the forward-facing branches of clinical pathology that sees patients. And predominantly, those are patients that need blood components. So we treat sickle cell disease patients and other patients who need various manipulations of their blood. So with that, pre-COVID, and so until March, I was not a virologist. I wasn't studying, obviously, COVID or anything having to do with COVID. And actually, 90% of my time was spent in an R01 funded research lab, not having much clinical responsibility. And I'll just tell you a little bit of what I was doing beforehand, because it is relevant for the rest of the talk. And so prior to COVID, I was studying clinically relevant issues in transfusion medicine from the perspective of iron biology. And I will give you one example of what that means. And I was a translational researcher. So I was, in the lab, we use everything from cell culture to mouse models to human studies. And so, for example, one of the things that interests me has to do with blood donor epidemiology. So it turns out in the United States, we need about 12 to 13 million units of blood a year, and only about 5 million Americans actually volunteer to donate blood. So most people who donate blood end up donating twice or 3 times a year to meet the demand for the blood supply. One of the consequences of that is that up to 66% of women and 50% of men who are altruistic, wonderful people who donate blood actually end up becoming iron deficient from their frequent blood donations. And we still don't know whether that's, in a sense, okay or not, and whether that has any adverse side effects to these people who are donating blood for society. And so after doing many animal studies to model this and to look at this, we were in the midst of a clinical trial where we were recruiting blood donors and testing them for iron deficiency. If they were iron deficient at baseline, we had them, and they could donate a unit of blood, so they met the criteria, the FDA criteria for donating blood. We actually had them donate blood, and then we randomized them in double-blind fashion to either a gram of iron or placebo IV, and that was enough to replete them to a point where, from an iron perspective, they're back up to where they were before they started donating. And then we follow them for a number of months and have them donate again and do various studies, neurocognitive testing, MRIs, fMRIs of the brain to look at cognition, to see whether this has any impact on anything, any side effects to these volunteers. Unfortunately, the study had to be stopped in March, and we're now starting up again. And so maybe, depending on how this talk goes, maybe I'll be invited back again in a year or so to give an update as to what the results are, because it's double blind, so I can't really look at the data. But these volunteers actually became useful for us developing serological testing, as I'll explain in a little bit. But unfortunately, March hit, and this is a more recent map of what the United States looks like in terms of incidence of COVID-19. I say New York was the epicenter. Unfortunately, think we were the epicenter. I don't think we'll remain the epicenter. But that's sort of where we are. And then everything came to a halt in March. And I have a large contingent of European postdocs, and we were told basically to shut down, stop everything. But I and they felt that if there's something we can contribute, that we should stay on and continue working throughout the March and April height of the pandemic in New York. And so I'll tell you about what we were doing, or some of what we were doing. So if you think back to March 1st was the index case in New York. Since then, a lot has happened. But back in March 1st, the focus was really on the molecular testing. So when I say molecular testing, what I mean is the nasal swabs, where you're looking for RNA of the virus. And that's done in a different lab than the one that I'm affiliated with. And I can tell you that the people who are in charge of that, Susan Whittier, Dan Green, Mahesh Mansukani, had many sleepless nights in March, bringing on the capacity for molecular testing at Columbia. Initially, the testing was only allowed to be done either at the CDC or the New York State Department of Health. And so there was a lot of confusion in the beginning, but there was a lot of work done there. And I won't be talking about that because I'm not expert on the molecular testing. But what I will be talking about is the antibody testing. And so somewhere around mid-March, we started hearing murmurs of people wanting antibody testing. And the murmuring came from the hospital epidemiologists, from the hospital leadership. And ultimately, we started having weekly meetings with the New York State Department of Health. And some of this also came down from the governor, who wanted us to begin antibody testing. And so what do I mean by antibody testing, or serologic testing? And so it basically measures the immune response or antibodies to infection. And it's in general meant to identify prevalence of disease. So how many people have had the disease as opposed to who has it right now. And we still don't know if this is the case or not, but it's useful to know whether someone is immune to the disease or not. And ultimately, from a societal perspective, if we have enough people who are immune, then we can assess whether we have herd immunity as a community. But why I became interested in it and why I decided to help bring up the antibody testing at Columbia was because ultimately I'm transfusion medicine trained, and one of the treatments in pandemic situations is use of convalescent plasma, which is taking plasma from people who have recovered, or antibodies that are extracted from people who are recovered, and trying to use them in people either who are sick or using it prophylactically in people who aren't sick yet, and to try to prevent disease in vulnerable populations. And so, I decided to focus on convalescent plasma. In order to get convalescent plasma donors, you have to have an antibody test. And at the time, we didn't have any in the United States. However, the market was flooded with what I would call minimally validated EUA assays. So just to be clear, none of the assays for antibodies that we currently have are FDA validated. They all have an emergency use authorization from the FDA, which means they haven't undergone the rigorous evaluation that you would have to go through to get real FDA approval. But they all have an EUA, and so we're allowed to use them clinically and report the results. And at the time in March, when I began becoming interested in this, there were 30 or 40 manufacturers, mostly from Asia, that were providing these assays, and we had no idea whether they were any good or not. And so if you look at the textbooks for what we think about how I guess the time course of a virus goes, there's an initial prodrome period, and then there's a period where you're going to be able to detect viral antigen, and in this case, since it's an RNA virus, viral RNA in the nose or in the lungs, if you can test there. And then eventually the immune system kicks in, and what's taught in the textbooks is that initially you get an IgM response, and then the B cells class switch, and then you get an IgG antibody response that one can detect serologically. And this can give you some sense for how, if you have an IgM response and not an IgG response, that will tell you that the infection was, within a week, and the longer you go, you should only be able to detect IgGs. that's what's taught in the textbooks. I'll show you some data that doesn't really hold up well for SARS-CoV-2. And so when we detect antibodies, what are we detecting antibodies to? And so this is a picture of coronavirus. You've probably seen them multiple times by now in talks, but for the most part, the immunodominant epitopes are thought to be in the nucleocapsid protein, which is involved in packaging the RNA, and in the spike glycoprotein, which is involved in the receptor adherence to the ACE2 receptor and getting the virus to infect epithelial cells. This is what's known from SARS-CoV-1 and from other coronaviruses. Those are thought to be the more immunodominant epitopes. And so in general, that's what we test for. And back in March, the only things that were really available were, you can see my mouse, but the things on the right, which look sort of like pregnancy tests. And they sort of work like pregnancy tests, and you put some blood on them and they tell you whether you're positive or negative. People were also developing more quantitative assays, which one format of them is called an ELISA, or an enzyme-linked immunosorbent assay. I'll show you some data because we ended up developing one of these as well. And so basically the way these work is you make serial dilutions of serum or plasma so that the blood gets more and more dilute as you go from left to right, and each row is a patient. And so somehow you react it in a well, and if you get a color reaction, the more color you have, the more antibodies the patient has. And so in this example here, patient A has no antibodies, patient B has antibodies, and at this point you were able to dilute them out. And this patient has, in row C, has more antibodies because it took a little longer to dilute them out. And so that's how those work. But at the time, these were not available. We only had these that were literally being thrown at me. I ended up evaluating about 5 of them. And they work sort of like pregnancy tests. You put a drop of plasma in a well, you add some saline that draws the antibodies through the paper by capillary action, and then inside of it there's some kind of COVID-19 antigen that's conjugated to something that produces a color like gold or something like that. If antibodies are present, they're going to bind the antigen, and then there's a secondary antibody sitting on a test strip, and that stops the conjugate, the complex, and then you can visualize it as a line. There's also a control line that tells you that the test was working. And so you know, we had 5 or 6 of these to evaluate to see whether they were any good. And the question becomes that, you know, how do you evaluate an assay if you don't have a gold standard? And so no one had any gold standard test to say, yes, this person has antibodies or not. And so how do you go about evaluating the test? And the way you do that is that, you know, you have to take a guess. And so what we knew is we had patients who were testing positive by PCR for the virus. And so we knew that they were infected. And we figured that if you wait 2 or so weeks, they would make antibodies. And so we had to find serum samples from patients who tested positive by nasal swab for virus. And so one of the first things I did in early March, and actually even in late February, was realize the importance of the 10,000 tubes of blood we throw out every day in the clinical labs, and quickly wrote an honest broker IRB to allow us to begin banking these samples for our clinical use in validating assays, but also for broader use in the Columbia community for other researchers who find these samples very valuable. And I can tell you that last I looked, you know, we're up to almost 25,000 serum samples that have been banked from, you know, over 5,000 unique patients that we're seeing at Columbia that are now available for people who want to do research on them. And if you do want to do research on them or want to get access to any of these samples, I just wanted to bring your attention that there is a website, in the Columbia website, that you can look up the biobank and then submit a request. And if it's reasonable and it is approved, then you'll get those samples to study. And just so that you're aware, in addition to blood, we also collected urine, CSF, feces, and P swabs, anything that we were throwing out that we thought would be valuable. We had a whole team of volunteers who were collecting them, coding them, and banking them for future use. And so those are available. And so now I had these serum samples at my disposal to validate the assays. And this is one of the tests we validated, and this is showing a positive result for IgM and IgG, and the control line is coming up positive. And so what do we focus on when we validate an assay as a clinical pathologist? And so for this type of serological assay, the most important thing is the sensitivity and specificity. So I apologize for being a little pedantic, but I think it's important for you to understand the difference between sensitivity, specificity, positive and negative predictive value when we think about lab tests. And so the sensitivity in Bayesian notation is the probability that you're going to test positive given that you have disease. And so what's the probability that the test comes up positive if someone was PCR positive 2 weeks prior? The specificity is the opposite question. What's the probability that it's going to be negative if the person doesn't have disease? Or in other words, was PCR negative by no swab? And so those are important characteristics that we need to calculate for any assay that we evaluate, because that tells us how well the test is going to be at picking up disease in the community. And so we took samples from our hospitalized patients. We knew that they were PCR positive. And as we broke it up into, we looked in their medical record to see when their onset of symptoms was. And so we had people that were still early on after onset of symptoms up to people who were 14 days or more after onset of symptoms. We only had 37 of these patients at the time. So remember, this is mid to late March. And so we still didn't have that much experience with the pandemic, so we didn't have that many patients yet who were that far out. And when we evaluated, I'm showing you data from the best pregnancy test sort of that we had. The sensitivity did go up the further out you were from onset of disease, but overall, if you use the IgM band or the IgG band, ultimately the best we got was a sensitivity of 57%. In other words, only 57% of people who tested positive by no swab were positive for antibodies 2 weeks out, which is okay, but it's not that great. When we did the opposite, when we took people who were PCR negative and we didn't expect them to have any antibodies, 95% of them, in fact, didn't have antibodies, but that means 5 out of 100 would be false positives. And so as a clinical pathologist, I'm interested in the sensitivity and specificity of the test because I want to know how good the test is. But from the user perspective, you want to know what the positive and negative predictive value is. And so it's sort of the opposite question in the sense that the positive predictive value is saying, if I know I've tested positive, what's the probability I have disease? So if you have a patient who tests positive or you yourself test positive, does that tell you whether you have the disease or not? And similarly with the negative predictive value, if I test negative, does that mean I don't have the disease? And so I just want to take a little bit of time it's a little nuanced, but it turns out that the positive and negative predictive value depend on the sensitivity and specificity, but they also depend on the prevalence of disease, which becomes important. So to show you how this works, we'll just do a little thought experiment. So imagine if you take this pregnancy test that's 50% sensitive and 95% specific, and you go to an island where no one has seen COVID-19 yet, and the prevalence of disease is 0%. So if you use this test on that population, since the specificity is 95%, 95 would be test negative and 5 would test positive, because a 95% specificity means you would get 5 false positives. So what that means in terms of its positive predictive value is that if you test positive on this island, that's a false positive. So the positive predictive value is 0% because the prevalence is 0. No one has disease on the island. And so the positive predictive value of getting a positive test is 0. On the other hand, the negative predictive value, if you get a negative test, since no one has disease on the island, is 100%. If you take the other extreme of, if you go to this island and everyone has had COVID-19, so you have 100 people that you're sure had COVID-19, Since the sensitivity is 57%, that means 57 of them would test positive and 43 would test negative. And so the positive predictive value here is actually 100%. If you test positive, you're 100% sure that you had the disease because, again, the prevalence is 100%. But if the test is negative, that tells you nothing because no one, everyone should have tested positive. And then if the prevalence is anywhere in between, then the positive and negative predictive values, range somewhere in between 0 to 100%, depending on the sensitivity and specificity of the test. And so we actually developed a tool that, if you go to this website, you could actually it actually plugs in for you the prevalence of disease over the last 7 days from what we've observed at Columbia. You can put in the sensitivity and specificity of the test, and it tells you what the positive and negative predictive value is. And so if you look at the best COVID pregnancy test sort of that we had, the positive predictive value, if you assume at the time that the prevalence was about 2%, the positive predictive value was only 19%. In other words, if you got a positive result on one of those little point of care tests, there was an 81% chance that that's a false positive. If you're going to use one of those tests right now, where the prevalence is probably somewhere around 25% based on what we're observing, your chance of a false positive is still 21%. So even though the specificity is 95%, and you may think it's a pretty good assay, your chance of getting a false positive is still 1 in 5 if you use one of these point of care tests these days, and which is why I'm actually happy. Early on, I was worried that the FDA would approve these for home use, and they actually pulled back from that, and these, for the most part, are not approved for home use because I think it would lead to a lot of confusion and mischaracterization of who has disease and who doesn't. So now we're literally on April Fool's Day, April 1st. We're sort of at the height of the peak. We've come to the conclusion that none of these lateral flow assays that we've evaluated is going to be good enough. And the big question is, do we wait for the commercial vendors to come through for us? So we have large commercial platforms in the labs, and that would be ideal to run it on those commercial platforms. Or do we try to develop a test ourselves? We were told at the time that the commercial vendors were about 2 months away from being able to provide a test, which is actually true because we didn't get our test up and running in the lab until June 1st, and so it did take about 2 months. And we were getting a lot of pressure from hospital leadership to bring up an antibody test that would work. And so, we ended up having to go ahead and do it. And why we were really hesitant to do it and why we really wanted to wait for a commercial assay was that, as clinical pathologists, what we know is that most of the errors that happen in lab testing happen pre-analytically, so before the sample even gets to analysis. And so one of the advantages of having a clinical analyzer, and this is a picture of one of the analyzers we have in the lab, is that a tube goes in on the front, it gets scanned and registered so that you know that the tube is in the lab. There's actually a centrifuge in here, so it spins the tube down to separate the plasma or the serum from the blood. And then it sends the tube directly to the analyzer, which draws the sample, tests it. The test is pretty quick. It's an 18-minute assay. It's high throughput because it can run multiple assays every minute. It provides reproducible results because they've been doing this for years, and they have factories where they can make really reproducible reagents for us to use. And it's relatively cheap. So even though CMS might charge, or we can charge, I think, $45 for an antibody test, Roche, which is the system we use, only charges us around $5 or $6 for the reagents for a single test. And then post-analytically, the results get reported and they go immediately to Epic without any human having to intervene. And the tube goes into a refrigerator that automatically gets stored and could be brought back if we need to retest. And so no human intervention needs to happen. If we validate our own assay, then we have to manually register it, spin it, make sure the correct sample gets put in the right well. Analytically, there are a lot of issues. It's a 4-hour assay. The throughput is not as good. we're not a factory, so how reproducible are we from plate to plate becomes an issue, and it's quite labor intensive because you actually need people to pipette and do work to do this, and so it's expensive, it's labor intensive. And then analytically on the other end, once we have the results, it doesn't automatically go into Epic. We need, by regulations, we actually need 2 people to review them to make sure we report the right result for the right patient. And so it creates a lot of work and headache. But we went ahead and we did it for various reasons, partly because we needed to screen convalescent plasma donors, and partly because it was necessary for because many clinicians wanted to know whether they've made antibodies or not. It became politically very important to bring this up. And so we had to make a decision as to which viral proteins to target. Based on the literature and the immunodominant peptides, we chose both the nucleocapsid and spike trimer to validate. But that leads to another problem, is that in order to test for antibodies, you need the viral antigen. So we literally had to grow the protein up and purify it in a way. And fortunately, we were able to collaborate with David Ho and Filippo Mancia, who's in the Department of Physiology, who has a protein purification lab, and they helped us purify a sufficient amount of protein to allow us to validate and then test, do the clinical testing that we did over those 2 months. And just to give you a sense for how the assay works, you grow up the viral protein, you coat plastic wells with it, and then you incubate the patient's serum in the wells. If the patient has antibodies, it's going to bind the protein that you coated, and then you come in with a secondary antibody that's going to bind the patient's antibody and change the color so that you can detect it. So the first question is, how do you set a cutoff? So we want to tell people whether they have antibodies or not. And so the fact that I've been working with blood donors over the past 10 years in New York City gave me basically a biorepository of serum samples from hundreds of healthy New York blood donors from the pre-COVID era. And so I used hundreds of these to test and come up with a cutoff that was 99.9% specific. In other words, only 1 out of 1,000 people from the pre-COVID era were positive based on the cutoffs we chose. And so we validated a spike IgG, IgM, and an IgG to the nucleocapsid proteins, at least initially. And here I'm just showing you data on the sensitivity. So now we have more samples to play with. On the x-axis, I'm showing you time since onset of symptoms from 0 to 6 weeks out. And on the y-axis, I'm just showing you the amount of color, the strength of the reaction in the ELISA. And what you can see is that early on you don't get much reactivity, but as people are further out from disease, you start to see positivity. This is for IgM, and surprisingly, the sensitivity for IgM early on was no better than IgG, and in fact, IgG was better, and this is now observed from multiple assays. Not really sure why this is, but we don't really see IgM antibodies coming up earlier than IgG using the available assays that people are using these days. And ultimately, the sensitivity was a little bit disappointing at first, somewhere around 87% at 3 weeks out. So in other words, 87% of people who were PCR positive were testing positive 3 weeks out. But we asked the question, why are these people who are below the cutoff and 10 or more days out from disease, why are they not positive for antibodies? And when we looked in the medical record, we realized that many of our patients are immunocompromised. And so these are examples of patients with marginal zone lymphoma or chronic lymphocytic leukemia on Rituxan, which you'd expect would kill your plasma cells, and so you wouldn't be able to mount an antibody response. You have patients with kidney transplant or methotrexate with no white cell count. You have aplastic anemia patients and people with Alzheimer's on various immunosuppressive agents. And so many of these patients that didn't make antibodies could be explained by the fact that our patient population is such that we see a lot of immunocompromised patients. And so the actual sensitivity when you remove many of these immunocompromised patients is much higher, and it's in the mid to high 90s. going to skip this for the sake of time. For the specificity, we also wanted to make sure that we didn't have cross-reactivity with any of the other coronaviruses that are endemic in New York. And so there are other coronaviruses that we see every year. We were able to get serum samples from them, and they were all negative. We did have a couple, or one really strong positive person who tested negative for COVID-19 by PCR. But when you look at her clinical history, she had increased work of breathing times 7 days. This was in March. So even though her COVID-19 PCR was negative, we're not 100% sure she didn't have COVID-19 maybe in late February or early March, and we just missed it. And so, we tested her, so we included her. And so, we're saying the specificity is 96%, but we're not 100% sure that she's really a false positive. We also had to set up quality control reagents to make sure that our assays work the same every day of testing. There are a lot of other things that go into a validation, but we were able to validate the test in about 2 weeks, and by mid-April, the New York State Department of Health gave us approval to begin testing our healthcare workers. And based on the prevalence, the positive predictive value was about 90%, the negative predictive value was about 98%. And this was, at the time, the best test that was available, and the hospital epidemiologist and hospital leadership felt that this was good enough to get going. And so we started testing, and June 1st, we were able to bring up the commercial platforms, and during that intervening period, we actually tested around 6,000, mostly healthcare workers, who wanted to get tested at Columbia for various reasons. And then when we did bring up the commercial test, that was a nervous moment for me, because we spent all this time validating the test and developing this test and growing up protein. How did we compare to the commercial platforms? And fortunately, just about every single sample that we were calling negative was also being called negative on the commercial platforms, and we looked at a number. And any sample that we called positive was also coming up positive on the commercial platform. So we felt pretty good that we were giving pretty accurate results for the 6,000 people who got tested before June and before we were using the commercial platforms. And for the most part, other than for research, where we are still running our laboratory developed ELISA, most of the clinical testing is now being performed on the commercial platforms, which again could be done 24/7 and without much human intervention. So now I have maybe 5 or 10 minutes. I'll just take you through some clinical pearls or clinical correlates, things that we learned as we were testing all these people. And so one of the first things that we did is we were actually collecting serum samples since late February. So I was taking serum samples from emergency room patients, both the psych emergency room, pediatric, and adult. They were de-identified. They were just randomly thrown out serum samples because we wanted to see at what point we could start detecting antibodies coming up in New York City. And we actually found 3 cases at Columbia from late February that were positive for antibodies. And if you do the modeling and the math with the different multiplicative numbers for how quickly the virus replicates in the community, and you do various modeling, it suggests that even though our index case was March 1st, the virus was probably already present in New York City by late January or early February. This is pending publication. It's not published yet, but this has now been published by other groups, and so it should come as no surprise that the virus was circulating much earlier than the index case. And I guess the only lesson to be learned here is that had we begun testing much earlier than we did, we potentially could have prevented this huge spike in cases in later March, because then we could have intervened a lot earlier and maybe shut down a week or 2 earlier, and that would have saved many lives. In terms of the convalescent plasma story, so again, the reason I was so interested in bringing up serological tests is because I'm a transfusion medicine physician, we wanted to do convalescent plasma studies at Columbia. So as many, over 15,000 units of plasma across the country were used for compassionate use, which means they were given not in the setting of a study. They were just given because there was nothing else to do, and we're not going to, in a sense, learn anything from that experience. But we wanted to support randomized controlled trials that would test whether convalescent plasma is beneficial, and if it is, in which situation it is. And so the idea there is you just give people plasma or antibodies from people who convalesced, and that should provide immediate but temporary immunity. It's been tried for other viruses, and actually currently if you get bitten by a rabid dog, the treatment for it is you're going to get hyperimmune globulin from rabies hyperimmune globulin, and you're going to get a vaccine against rabies to prevent because it's almost invariably lethal if you get rabies, and that gives you a pretty good outcome. And so it's pretty effective for many viruses, and it's been tried in other epidemics, although it's very difficult to do randomized controlled trials in the midst of a pandemic, and invariably for all these prior uses, we don't really have good data on whether it's effective or not. There are hints that it may be effective, but we don't have any definitive evidence. And so recently in JAMA, a report came out from China where they did use convalescent plasma on either patients with severe COVID or life-threatening COVID disease. Again, difficult to do a randomized controlled trial in the midst of a pandemic. They were able to randomize 45 people with severe COVID disease, and if you look at that subgroup, there seemed to be a benefit to getting convalescent plasma in terms of overall survival. It wasn't borne out in those with life-threatening disease or overall, but at least in that subgroup, there was a hint that there may be some benefit. And we initiated a similar study. This was with Ian Lipkin and Max O'Donnell in the ICU, and various others. And my role in that was to help in screening the convalescent plasma donors to try to get plasma in our blood bank to support the trial. And that's why we brought up the ELISA assay to qualify donors. And over the past, since March, we've screened close to 500 subjects who came into Columbia to be tested. Whenever I do studies in blood donors, females tend to come out more than males, even though males are affected by COVID-19 more than females. And in general, they were screened, the pretest probability was much higher in them because they were screened to likely have had COVID-19, and so about 60% of them were testing positive for COVID-19. And just to give you the current status of the trial, we've thus far randomized 60 subjects. Many people ask, so what does the data show? Unfortunately, when you do a blinded randomized trial, it's not so straightforward to look at the data. 60 is well below what we need or what we powered the study for. And so every time you look at the data, it does affect your chance for a type 1 error. And so it affects the alpha or the p-value that you ultimately report. And so it's dangerous to look at the data unless you're ready to do so. And so we have not formally looked at the data yet. And unfortunately, we're now over here where we have very few cases in New York. Well, actually, fortunately, we're over here in New York where we have very few cases. And so it's very difficult to recruit right now for this study. And so we might get one subject randomized a week if we're lucky. And what we're actually doing this week is transferring a lot of the plasma we collected down to Alabama, to another site, and to Brazil, where there's another site that has IRB approval now, to basically continue our study and try to use the plasma we collected here so that it's the same plasma, and so that we can combine the data and do the same study in Brazil and Alabama to try to increase our numbers to try to get us the definitive evidence we need of whether convalescent plasma might be helpful in ICU patients. We're also working on bringing up hyperimmune globulin, which is antibodies extracted from convalescent plasma, and we're hoping to use that for at least initially for prophylactic use for people who are very vulnerable, such as those with cancer and chemotherapy who basically don't have an immune system, and so that even if you vaccinated them, that wouldn't protect them. And so the thought there being giving them passive antibodies to try to protect them for their chemotherapy. So just another sort of clinical correlate or pearl that we learned. And so these are viral titers antibody titers that we did on the convalescent plasma donors, and you can see that there's a range of reactivity. This is the dilution, so how far you have to dilute the plasma before you lose reactivity. These are patients who we have in our hospital who have respiratory distress who are in the ICU, and hopefully you can appreciate that the curve is shifted to the right, so they have much more antibodies actually than the convalescent plasma donors who for the most part weren't hospitalized. These are people who had COVID-19 and stayed at home, recovered, and then came in to be screened in our clinic. This third population is the children with multi-inflammatory syndrome that we now have close to 50 at Columbia, and they tend to have lower antibodies than the ICU patient, the adult ICU patients that we see. And we've also looked at their neutralization titers, which is another test that I didn't speak to you about, but another test that we and others have brought up is not only looking at whether they have antibodies, but also looking at whether the antibodies can neutralize live virus or pseudovirus and prevent the virus from infecting cells. And it turns out that the kids with the MIS, with the multi-inflammatory syndrome, may have a defect, and their neutralization titers are much lower than the ICU patients and the convalescent plasma donors who recovered at home. To take it to another extreme, we also have samples from people who died and succumbed to infection, and those are the autopsy cases. And so you can see here that they actually have the highest antibody titers of anyone. And so what seems to happen is that the more sick you are, the higher the antibodies are. And so having antibodies doesn't necessarily mean you're protected, because clearly these people have high degree of antibodies and they're neutralizing, and it clearly didn't protect them. And so there's more to just the antibodies. And simply having antibodies may not tell the whole story. I think one final curiosity or something that came out of our screenings is that out of the 500 people that we screened in our convalescent plasma donor, and this I think will be the last story I'll tell you, as part of the study, because they're blood donors, we did collect information on address, and so we knew that there were among them about 34 cohabitants. And so for the most part, they're husband and wives or people who live together, both of whom thought they had COVID-19 symptoms or tested positive for COVID-19. And when we tested them for antibodies, interestingly, 4 couples didn't have antibodies in either. 16 couples both tested positive, but in 14, which is roughly 40% of them, they were discordant, where they both had COVID-19 or likely had COVID-19, but one is testing positive for antibodies and the other is completely negative. We now see this a lot in family studies, and I've even found that I have a set of monozygotic twins, 2 girls who are 12 years old, one of whom tests positive for antibodies, the other does not, and they both clearly had COVID-19. So why that is still a mystery. We're still trying to figure it out, and we're hoping to begin large-scale family studies to try to get at why that might be. And part of the story might be that you also might have T cells that might help you with the virus, and so people who have a good T cell response may not need the antibodies. So some remaining questions that I'm left with, and I'll leave you with. So we see many subjects who are PCR positive, clearly have had COVID-19, and are serologically negative. Even more so, we see many people who were symptomatic, who want to know whether they truly had COVID-19 or not. They stayed home, they were never PCR'd, so you don't know for sure whether they've had it or not. And they're testing serologically negative. About 60% of people who had pretty suspicious symptoms and are tested serologically are testing negative. We don't know why that is. I mean, it could be that you really didn't have COVID-19. It could be that you had it, but you didn't need to make antibodies to clear it. It could be that you made a low level of antibodies which were sufficient to clear it, but the assay sensitivity is just too low. Could be that we're not making the viral epitopes we're using in our testing is not the correct ones, and you've made antibodies to something else in the virus, the membrane protein or the envelope protein, which we're now testing for. Could be that you're making IgA antibodies, which are in the mucosa that we might not be testing for, and you might not, I would argue that it might be more important for you to have IgA antibodies in your mucous membranes or in your saliva when you're fighting a respiratory virus, as opposed to having antibodies in your bloodstream. And it could just be that we're getting the timing wrong. Maybe if we waited longer or we tested you earlier, we would have captured an antibody response that now has disappeared. I still don't know the answer to the question. We're working on it. The billion-dollar question is really, do the antibodies provide you with lasting immunity? And if so, how long does that last for? Still don't know the answer to that. I'm sorry, I can't give you the answer or any thoughts on it. We still truly don't know. And then I still truly don't know will passive immune therapy work. And so there are people still working on convalescent plasma, hyperimmune globulin, and now there are trials with monoclonal antibodies that are underway. And so we still don't know to what extent these antibodies will work and in which populations they're going to work for. Finally, I'll end with acknowledgments. This was a collaborative effort with many people. I have to thank David Ho and his group in the Aaron Diamond's AIDS Research Center, who really helped bring up the ELISA testing. Filippo Mancha and his group in the Department of Physiology, who helped us purify all those proteins that we needed. And then in our department, headed by Kevin Roth and Steve Spitanik, who's head of the labs, who've been my mentors throughout. Francesca LaCarpia, who's a postdoc in the lab who did a lot of the work, along with Wendy Lin and Sandeep Wantakal, who are fellows, who really did a bunch of the work, and many of the pathology residents who helped look up data in the medical record so that we would get a sense for when the onset of symptoms were when we calculated our sensitivity. And then there's a whole group of postdocs through the CRAC team and led by Ken Olive that volunteered in the lab. So we were running a COVID lab that had anywhere between 5 to 15 volunteers a day who were working on biorepository work, developing ELISA, and then testing the 6,000 healthcare workers that we tested between April and June. So I just need to acknowledge all the people who did the work. And with that, I'm open for questions.
Well, thank you so much, Dr. Ho. That was incredibly informative. And I think I can speak for myself, but I think it's true of most of us that we see the lab results when they get printed out on Epic or some other format, but have no sense really of what goes into it on the back end. And so it's great to be enlightened about that. I also really wanted to thank you. I'm sure that the past few months have been incredibly busy and pressured for you and your whole team, and so really appreciate you making the time to talk with us.
No, it's my pleasure, and I think it's one of the first times that people were asking clinical pathologists to speak up, so I'm happy to do so.
So we've had a bunch of questions come in. So I'll start with one of them. So you nicely described the poor sensitivity and predictive value of the point of care assessments or tests, and wondering, hindsight is perfect, of course, but in retrospect, do you think that it was a mistake by the FDA to let these tests go out into the public?
So I think there's been a lot of mistakes that have been made by the FDA, and I do think it was a mistake. I'm glad that they didn't allow the point of care test to really go out into the public. So they weren't allowed to market to the point of care. They were allowed to market to labs, which, if they're doing things correctly, they would validate the test and come to the realization that for the most part they aren't the best assays, and they are. I won't say they're not useful. There are situations where they are useful, but you just have to have a good understanding for how to use them. Where I think the FDA and BARDA really made a mistake, which really hurt us, is the whole convalescent plasma story. If I may go on a tangent a little bit, in that early on they decided, without any clinical evidence that convalescent plasma would work, through a large BARDA grant, that they would make convalescent plasma available to any hospital for free to be used for compassionate use. And that really hurt the randomized controlled trials, because now everyone was using it sort of willy-nilly, and 15,000 units got used, and we learned nothing as to whether it's effective or not. It's sort of like the hydroxychloroquine story. And so, I think that was a big mistake, and had they waited a little longer and allowed more places to do randomized controlled trials and actually get evidence for where it might be effective and where it might be harmful, that would have benefited society, and we wouldn't have been stuck now with only 60 subjects in a single center study, and we may have had more data to do a meta-analysis on.
So another question has to do with the approach that you used in validating your serologic test. So you compare the serum samples from individuals where they're PCR positive, and then their serum later. But late in your talk, you showed that the discordant rate was actually surprisingly high. Does that make you concerned about the sensitivity or the validity of the test that you developed?
So just to be clear, I think there's difference between analytical sensitivity and in a sense, biological sensitivity. So, when we validate the test, I need to make sure that I'm testing someone who for sure had COVID-19. And if you know someone for sure has had COVID-19 and was sick enough to be hospitalized or sick enough to come in, put themselves at risk coming in to the tent to get screened, then of those people, then it's highly sensitive. Where the problem becomes where the test is not as sensitive, and it's not just our test, it's all the tests, is if you're looking at people who stayed home and never got tested. So if you weren't sick enough to get tested, then the sensitivity, so we don't know for sure whether you've had it or not, right? So you never got tested, you had suspicious symptoms, you had anosmia, what else could it be? But you never got tested, so we don't know for sure. Of those people, most people are testing negative, and that it's very disappointing, I know, to many people. But that might be a biological thing. It's not that our test doesn't pick up the antibodies, because it clearly picks up the antibodies. But the people who weren't sick enough aren't making a high enough antibody titer for us to detect, or not making making different kinds of antibodies, or something is different, or you're clearing the virus using T cells or a cell-mediated response, and so you're not required to make a high, serologic response, and so we're just not seeing it. And I don't know the answer to the question, and I don't know for sure that it's not that our assay is not sensitive enough to pick out those small antibodies. But if you make the assay very sensitive to pick out very low level of antibodies, you'll start getting false positives a lot, where people who had other coronaviruses or other viruses, you'll start getting false positives, and so you have to be very careful when you set the threshold for what you call negative and positive. Does that make sense?
Yep, absolutely. That was very clear. Thank you. Another question is, can you comment on how T cell responses figure into recovery, and is there any work being done to integrate B cell and T cell responses?
Yeah, no, so it's not just in our lab. I know there are many people across the country who are working on that. And so we have a cohort of subjects who are discordant, where we're pretty sure both have had disease, but one has made antibodies, one hasn't. And we actually have peripheral blood monocytes stored on them. And so yes, now it's the work of thawing them, growing up the T cells, and seeing whether there's a T cell response, and seeing whether that can explain the discordance. And we're planning. It's still difficult to plan and hire coordinators and do studies in this. We're still, in a sense, in the midst of a pandemic. But we are planning community family studies where we will be able to collect you need peripheral blood monocytes, and you need to store them in a way that you can grow them up. And the other challenge there is that If there happens to be any virus in that sample, if you start growing it up in an incubator, then you might grow up the virus, and so there's an EH&S risk, and so you have to do some testing or do it in a BSL-3 facility. So there are limitations to what we can do, but we are planning on doing those studies, and I'm aware of many other groups who are doing them concurrently. We'll see what comes out. There are also potentially genetic factors that can explain it too. So there's MHC restriction, there's the whole ABO story, which we're still not sure about, whether type O is protective or not. And there are people within a family who may be A and O, depending on the blood types of the parents. And so there are potentially other explanations for why people are discordant. And if I can add one other thought, the way you get exposed to the virus might make a difference too. So, if you ingest the virus and you get the virus orally as opposed to inhaling it, and the dose that you get may make a difference as well as to whether you get severely sick or not. So if you get a very small dose and you ingest it orally, maybe it's not as bad as if you get a large dose that you inhale deep into your lungs.
Okay. In regards to the use of convalescent plasma, how important do you expect it will be in regards to the timing of when that treatment is given, the timing and the course of the illness?
So if you look at prior uses of convalescent plasma and hyperimmune globulin, in general, the earlier you give it, the better the outcomes. And so, the reason we went forward with the ICU study first is because we saw that everyone was using it for compassionate use, and they were using it in the ICU. And so we wanted to provide some evidence for or against that use, because people were, in a sense, wasting convalescent plasma in ICU settings where we didn't know if it was harmful or beneficial. So that's why we targeted that group for the initial randomized control trial. But we did have hope to do other trials for more prophylactic use where we think it might be more effective. And as I said, we're now evaluating hyperimmune globulin products, which are, in a sense, a little better. So if you take liters and liters of convalescent plasma, pool it, virally inactivate it, and extract out the antibodies, you can actually put them in little vials and give people IM shots or intramuscular shots of antibodies that might be protective. And so you could think about going into nursing homes or like I said, vulnerable populations. We're hoping to do it in cancer patients to see if it can be effective in preventing disease in those settings.
Great. In the COVID-related biobank that you mentioned that Columbia has, how is the consent process addressed?
So we're the honest broker. And so there's an honest broker procedure. And so you would get de-identified specimens. And so there's a consent waiver if you're going to get de-identified specimens and with limited information given so that you couldn't figure out who the patient was. But there are also consented biobanks now too. And so there are various ways to get samples. And some of these patients were retrospectively consented, so that there's a cohort that was retrospectively consented, so that to allow people to do DNA studies and other types of studies that you would need consent for.
Great. I think our last question is, you really nicely demonstrated how the prevalence of a condition can determine the predictive value of the diagnostic measures. In something as dynamic as the COVID pandemic, how do you think about getting accurate prevalence numbers?
Yeah, so I showed a link at the bottom of that slide where I showed that calculator. That link was developed by our IT people in our department. That actually captures the seroprevalence from the last 7 days based on the testing we've done. And so you could use that as an estimate for what the current seroprevalence is. And if you track it, you can actually see how it increases. Obviously, there's a pretest probability issue. So early on, when we were just testing people who were symptomatic or who had, a positive diagnosis, our seroprevalence was higher. Now, where we opened it up and any healthcare worker can be tested, it's more of a random test of the population. And people have gone out to supermarkets and to test various people, and there are now reports of seroprevalence in New York City, and it's variable. It's somewhere, I've seen numbers anywhere from 20% to 40%. But yes, it clearly does, play a role in how good the test is, or how you interpret the test.
Okay, I think that's it for our questions. And again, I just really wanted to thank you for taking the time and giving us such an informative talk. Really appreciate it. And thanks everyone for listening to Grand Rounds, and we will meet again in a week.
Thank you.
All right. Bye-bye.